This is part 4 of chapter 9. Uh, so in this last section, I wanted to talk about fermentation and talk just very briefly about how some of these enzymatic reactions that are part of these energy harvesting reactions can be regulated. Um, so as we discussed earlier in this chapter, without oxygen, most of these different pathways that we've been talking about won't work. So glycolysis can take place in the absence of oxygen, um, but if we don't have oxygen present, we can't really get our pyruvate that would be produced by glycolysis into the matrix of the mitochondria. That would obviously prevent the citric acid cycle from functioning properly. Um, but most importantly, without oxygen, right? We can't pull those electrons out of the end of the electron transport chain, and so we get this just traffic jam of electrons in that chain, and the chain stops working and stops pumping protons, and so then ATP synthase stops working, and we're just not making the ATP that the cell needs anymore. So bottom line is, without oxygen, we can carry out glycolysis, but that's about it. And you may remember glycolysis will produce 2 ATP, right? So that's better than nothing, but it certainly doesn't compare to the 32 or 34 ATP that you're going to get through full aerobic respiration. And so, how does this tie in with fermentation? Well, the issue is, even with glycolysis, is if there's no oxygen around, um, the cell will basically run out of NAD plus, so oxidized electron carriers. This is a very low abundance molecule. Cells just don't have a lot of it on hand, and they don't like to make a lot of it for whatever reason. So you have a very limited supply of NAD plus in your cells. Um, but for glycolysis to happen, you need some of this NAD plus, right? Because some of the key steps in glycolysis are taking electrons from glucose and giving them to NAD plus to produce NADH. Okay, and normally this isn't a problem, this low abundance, because what happens is the NADH that's produced goes off to the electron transport chain, right, in the mitochondria, drops off its electrons, and gets converted back into NAD plus. So we have this constant recycling that happens. So even though you don't have a lot of NAD plus in your cells, you have enough to keep glycolysis going. But now, if we don't have oxygen around and the electron transport chain is shut down, NADH can't drop off its electrons anymore. So if the electron transport chain's backed up, it just won't accept any new electrons at all. Uh, so pretty quickly, what would happen is, even in the absence of oxygen, your cell's cranking through glycolysis still, but eventually it uses up all the NAD plus, and then at that point, even glycolysis would stop. So, and if glycolysis stops now, your cells are making 0 ATP, right? And they're gonna be in big trouble. Cells are gonna start dying quickly. So the whole point of fermentation is to give NADH molecules somewhere else to deposit their electrons so that they can be recycled back to NAD plus, and then we can keep glycolysis happening. So essentially, fermentation is just a way to allow glycolysis to keep occurring when the electron transport chain is shut down in the absence of oxygen. And so what this looks like depends on what type of cell we're talking about. There's slight differences whether you're talking about like yeast or plant cells versus an animal cell. What we're looking at here is the situation in animal cells. This would be what happens in our cells. Um, and the way this works is, instead of NADH giving its electrons to the electron transport chain, NADH will give its electrons directly to pyruvate, which, remember, is one of the main products of glycolysis. And so when we do that, the the pyruvate is going to be converted into this molecule called lactic acid or lactate, right? And so you're familiar with this idea. You're at the gym running on the treadmill or whatever you do, some aerobic exercise, and you're just, you're breathing heavy and you're not getting enough oxygen to those muscle cells that are using lots of ATP. Some of those cells end up in an anaerobic state, right? They don't have enough oxygen. Electron transport chains start shutting down, and so your muscle cells switch into fermentation mode, and they're going to start producing lactic acid just as a byproduct. And the lactic acid builds up, and your muscles start burning, and feel like you want to get off the treadmill, right? Stop running. So just to be clear, right, this lactic acid is just a byproduct. So your cells, your body doesn't really have any use for that, right? It's just kind of an unfortunate byproduct of fermentation. And again, the whole point of fermentation is just to keep glycolysis happening so we can keep making those 2 ATP, which will hopefully keep your muscle cells alive until you get off the treadmill and you're able to catch up on your breathing and get enough oxygen to those cells. If we're talking about yeast cells, it's basically the same idea, but there's one extra step. So instead of the NADH giving its electrons to pyruvate directly, pyruvate gets converted in this enzymatic reaction into this molecule called acetaldehyde, and then NADH gives its electrons to acetaldehyde, which converts it into ethanol, right? And so, this is the basis of producing, all the alcoholic beverages that people enjoy, right? Wine and beer and all that good stuff, right? So you're basically putting yeast in the presence of some sugar source, and then depriving it of oxygen, so it starts to carry out this fermentation process, and ethanol will be produced just as a byproduct. All right, so this is just kind of the big picture summary again. So just to remind you, right, we started out this chapter talking about glycolysis and how that the end of glycolysis serves as this fork in the metabolic road, so to speak, where if we have oxygen present, we continue on to pyruvate oxidation in the mitochondria, citric acid cycle, electron transport chain, and we make a ton of ATP. Whereas if we don't have oxygen present, we're stuck basically just repeating glycolysis through this process of fermentation, and we can at least get a couple of ATP being made, and hopefully that'll keep our cells alive for the short term. But it's not really a long-term solution. So, best case scenario, we're usually getting, if we've got oxygen present, somewhere between 30 and 32 ATP. Sometimes you have to use a couple of ATP to get the pyruvate into the mitochondria. So that's why it's a little bit variable. All right, so again, thinking of this big picture, I just wanted to plant the seed in the back of your brain to get you thinking about the fact that these pathways that we have been talking about here in terms of these energy concepts are also connected to other metabolic pathways in your cells. You might remember in the last chapter I showed you an image that was had a bunch of these crazy colored lines all interwoven together that was trying to point out the fact that your cell's metabolism is complicated and interconnected. So I just want to re-emphasize that. So here's kind of another version of that same idea, just focused on some of these energy pathways that we were just talking about: carbohydrate metabolism, right, glycolysis to pyruvate oxidation, etc. And so, you don't have to memorize this, but what you see here is, other metabolic pathways, right? Lipid metabolism, nucleic acid metabolism, either feeding into or out of some of these pathways that we just discussed. And so what does that mean? Well, that means, there could be some molecule produced during lipid metabolism that can actually be used in glycolysis in some way, or vice versa, right? Some molecule that's an intermediate in one of those 10 reactions in glycolysis could be pulled out and used to help generate certain types of lipids. So just keep that in mind, right? All these different metabolic pathways are interconnected, which makes it a little bit complicated and overwhelming to think about. But people have been studying these things for a long time and actually have a pretty good handle on how all this stuff fits together. Yeah, so here's just a few examples of some of those what we would call interconversions, right? You know, things from carbohydrate metabolism being used in lipid pathways or protein pathways or vice versa. And so, I'm not going to test you on the details here, so I won't spend a lot of time on it, but I just want you to be aware that is the case. So those were catabolic interconversions. So we're basically talking about molecules that were being produced as a result of breaking things down, right? Catabolism is breaking things down. Anabolic interconversions would be as a result of building up larger molecules. And I did want to point out that these pathways that we've just been talking about, which were basically all involved with breaking glucose down, right? Glycolysis, citric acid cycle, all these things were kind of breaking down glucose into smaller parts and stripping some of that energy out. These pathways can be run in reverse, so we could run glycolysis backwards. It would require an input of energy, but we could do that to actually produce new glucose. This process called gluconeogenesis. And so, this is going to be less frequent, but you could imagine, there might be situations where cells need to or want to produce glucose. They've got an abundance of energy. And so, instead of reinventing the wheel, we're just going to use these pathways that already exist and just run them backwards. And lastly here, I wanted to tie this back in with our discussions in Chapter 8 of enzyme regulation. And you might remember we talked briefly about allosteric enzyme regulation. So allosterically regulated enzymes were enzymes that usually have a quaternary structure, and they could switch between active and inactive conformations depending on whether an inhibitor or an activator molecule was present. And one of the reasons I brought up that allosteric type of regulation is because there are some key allosteric enzymes in some of these pathways in Chapter 9. So for example, in the middle of glycolysis, there's an enzyme called phosphofructokinase that is an allosterically regulated enzyme. And so how does that work? What are its activators or inhibitors? What you can see here is, interestingly, it's molecules that would kind of make sense acting as the regulators. So, for example, if there's just a ton of ATP already present in the cell, right, the cell's been carrying out glycolysis and oxidative phosphorylation, and it's just produced an abundance of ATP. Doesn't really need any more ATP at that time. The ATP can actually bind to phosphofructokinase and switch it to the inactive conformation. So then we shut down glycolysis and citric acid cycle and everything downstream until ATP levels drop again, and we need to turn it back on. Whereas if the levels of ADP or AMP, monophosphate, in the cell are really high, those can bind to and activate that enzyme. Similarly, in the citric acid cycle, there's a couple of key enzymes here, citrate synthase, isocitrate dehydrogenase, that are similarly allosterically regulated. So, and by similar molecules, right? ATP or large amounts of NADH would inhibit ADP or large amounts of NAD plus would stimulate those enzymes. So it's just kind of a nice housekeeping method by which the cell can kind of automatically ramp up or ramp down these pathways depending on energy needs at that time. So that's it. Lots of information there in Chapter 9, but again, we're really focused on the overview, so don't sweat the details in terms of all those chemical structures and names of enzymes. You should really just focus on bigger picture and how all these different pieces that we've talked about fit together.